Hey everyone, Azim here. We are in chapter 19, the nervous system, continuing the nervous system. Specifically, we're going to learn more about general senses and special senses. We've already learned a little bit about the sensory system, sensory portion of the nervous system. We've learned about some basic receptor types. Remember, we learned this all the way even back in skin. Merkel cells, Meissner's corpuscles, Pacinian corpuscles. We're going to look in more detail of certain receptors, and in the next two videos after this one, we'll go in more detail about the eye and the ear. Since we're looking at the sensory aspect of the nervous system, it'll be really important to look at different receptors and what they're structured like and how their structure relates to their function. There are many different receptors for the many different senses we have. We have more than 5 senses. We have 6 or so conscious senses, maybe even more than that. But we have a lot of different senses. We sense things like vision. We sense things like things touching us. We sense the state of our body, and there's lots of different states of our body, how much oxygen there's in our blood, our temperature, how stretched out an organ is. And again, like I said, we'll focus on different parts of the ear, for hearing, for balance, and for we'll study different parts of the eye to look at how we see things. It'll be important to identify gross anatomy things as well as histology. So we'll be looking at some receptors and tissues under the microscope. I keep mentioning receptors. We've looked at receptors before, but to really drive home the point of what a receptor does, it might help to look at some examples that aren't necessarily rooted in biology. For example, this first picture here, we have a guy who's on the beach with a metal detector. Whoops, can't spell. Detector. A metal detector, I don't know exactly how it works. I think it has to do with magnets or electricity. It makes sense if you're going to detect metal. So let's say the stimulus for detection, in order for this metal detector to detect something, it has to detect metal. So the stimulus is metal, maybe magnets. It's not going to detect something that isn't metallic, or maybe not even magnetic. Probably has to generate some kind of current. Anyways, that's generally what a metal detector does. It works by detecting some property of metal. It doesn't detect other things. Here, what they're doing to this person, they are giving this person a lie detector test. Now, when you detect lies, I mean, there's no 100% way to really detect a lie, but we can infer lie detection from some physiological tests. For instance, they're measuring pulse here. They're measuring blood pressure. They might be measuring sweat. Let me put blood pressure and heart rate. They could be detecting sweat. Those are different stimuli that we can use to detect whether or not a person is lying. Again, not 100%, but those are the things that we measure. Those are stimuli. The smoke detector detects smoke. So you're detecting particles in the air. Each one of these detectors detects a very specific stimulus. Not everything will make these devices activate or set off. Metal detectors only detect anything metallic. Lie detectors only detect increases in blood pressure, increases in heart rate, increase in sweat production. Smoke detectors only detect particles in the air. These are specially designed detectors, or another word for a detector is a receptor, to detect specific things. Just like these specific things detect different stimuli, we have receptors that detect different stimuli. First, let's go over the different shapes, different structures of receptors. So we can categorize receptors based on structure. This first structural type is a free nerve ending. What that means is you have bare dendrites. Sensory neuron dendrites with no specializations. If I were to draw this in my style, dendrites, axon, cell body off to the side, this would be a pseudo-unipolar neuron, axon terminal. There's our simplified drawing of a neuron. And those free nerve endings are those bare dendrites. What kind of receptors can we find with free nerve endings? We have nociceptors. That's for detecting pain. Really, you're detecting damage, and our body perceives that as pain. Tissue damage. Thermoreceptors, you're detecting temperature. Root hair plexus, those nerve endings around the bulb of a hair, that's hair movement. And then baroreceptors, which we're going to go into more detail in a second. This is detecting pressure or stretch. We've talked about Pacinian corpuscles for pressure. That's specifically pressure in the skin. Baroreceptors are pressure and stretch in organs. So this is a visceral sensory receptor. Sometimes these receptors are unmyelinated, these axons. Sometimes they're unmyelinated, sometimes they're myelinated. A simple example is if you feel pain, there's different types of pain, but generally there's a slow, dull pain and a fast, sharp pain. I'm sure you felt like either a stabbing pain versus like this low, dull ache. Those low, dull aches are being transmitted along axons that are unmyelinated. Sharp pains are along myelinated axons. When you feel warmth, that's also tends to come in creep in slowly and itch as well. So those are unmyelinated neurons. Free nerve endings, bare dendrites. By having bare dendrites, they're easily exposed to stimuli like damage, hair movement, and changes in temperature and pressure stretch. More on baroreceptors. They line internal organs. Again, that means they're visceral sensory. They're really important to regulate the autonomic system. Autonomic is on the efferent side. Baroreceptors are on the afferent side. So this is part of that pathway. Let's say you have in your carotid sinus, the carotid artery is a blood vessel. Here in your carotid artery, you have a neuron that detects stretch. You're detecting blood pressure, for instance. That blood pressure receptor sends information to your medulla oblongata in your brain. And then your medulla oblongata decides, is my blood pressure okay? Should we tell the heart to decrease its rate in squeezing? Should we increase its rate in squeezing? We can adjust that if needed to maintain homeostasis. So we're regulating autonomic functions here. For most of these organs, like the aorta, which is another artery, the lungs, the digestive tract, your colon, which is your intestines, your urinary bladder, for most of these organs, the input is coming through the vagus nerve. Remember, vagus nerve is a mixed nerve. It's both sensory and motor. So sensory information is going along the vagus nerve. For the carotid sinus, that's with cranial nerve 9, glossopharyngeal. Some receptors have something more complex around the dendrites. If I were to draw my simplified neuron, here's my pseudo-unipolar neuron. Around the dendrites, we have connective tissue. And that connective tissue serves to modify the type of stimulus that's incoming. It modifies how the dendrites receive that information. The example that I believe I've given before is that, like, let's say your hand is a receptor, your hand is dendrites. If you poke your hand with something sharp, you're going to feel that immediately. It's going to feel very sharp. If you wrap your hand in a towel and then poke again with the same thing, you're going to feel the push, but it's going to feel very different. It's not going to feel very sharp. You'll just feel a dull push. Covering your dendrites with connective tissue changes the quality of information that comes in from that dendrite. So it's an adaptation in this case for detection of vibration and pressure in the skin and in muscles. We've seen these before. Pacinian corpuscles, Meissner's corpuscles, Ruffini corpuscles. Ruffini is for stretch, Pacinian is for pressure, Meissner's is light flutter and grip. Muscle spindles is for muscle stretch, and Golgi tendon organs is for tendon stretch. Might help if I write all this down. So you can see that we're detecting stretch and pressure, but this is for skin and muscles, not for organs. For organs, those are bare receptors, those are bare dendrites, free nerve endings. Deep pressure, okay. Great, so those are examples of complex neural receptors. You have the connective tissue around the dendrites to modify the stimulus that's being detected. Something that is especially true for these complex neural receptors, because they're in your muscles and skin, you can create a sort of map. We've talked about that homunculus in our primary somatosensory cortex. How does that map actually work? Let me attempt to draw a brain here. All right, here is a brain in a coronal frontal section, sort of. And if you recall what the homunculus looks like for the primary somatosensory cortex, the head region tends to be really big because the face is very sensitive. The torso is not that big. Hands are really big because hands are really sensitive. Legs aren't that big because they're less sensitive. And then feet are decently big as well. Should draw that head bigger. There we go. Okay, there's a attempt at a homunculus. So the hands, for instance, are very sensitive. The back and the legs are less sensitive because what that means is there are fewer receptors. And what that deals with is how much convergence of sensory neurons there is in this pathway. There's convergence of somatosensory receptors in some cases to create a large receptive field. What does that mean? Let's say that we're somewhere on the back, on your dorsum. Your back is not very sensitive. Low sensitivity equates with large receptive fields. How this happens is that if this is the skin on your back, you have this green receptor detecting from this region, should use green. This receptor in yellow is sensing from this area. And then this receptor in purple is sensing from this area. So each receptor is detecting from a different area of the skin. These are all first order sensory neurons. Cell bodies in the dorsal root ganglion or cranial ganglion. And then the second order sensory neuron is either going to be in the dorsal horn or medulla oblongata. What you'll notice here is that these, in this example, these three sensory neurons, the three first order sensory neurons, converge onto only one second order sensory neuron. So they're all sending information to the same neuron. So no matter which one you activate, the green one, the yellow one, or the purple one, you're only going to activate the same one sensory neuron. And that one sensory neuron will go to the thalamus, and then from the thalamus, it'll go to the primary somatosensory cortex. So fewer neurons, many neurons leading to fewer neurons. There's convergence. And when there's convergence, there's less area taken in your brain, and therefore low sensitivity is perceived. Large receptive field, more convergence, lower sensitivity. A test you can do to yourself, or really helps if someone does it to you, you could do something called a 2-point discrimination test. Take a compass or two sharp but not too sharp objects, and with your own eyes closed, have the person poke you with those 2 points or 1 point randomly, and you can test, like, do you report being poked with one thing or with two things? If you can distinguish between those two pokes, then, and in many different instances, then you pass that 2-point discrimination test. But like, if you poke me with two things back on the back of my neck, and the two things are too close, like they're close, maybe just a few millimeters apart, if you poke yourself, take a look up here, if you poke yourself within that receptive field, it's going to feel like 1 point. Even though you're getting poked with two things, it'll feel like one thing. So there's no 2-point discrimination in large receptive fields. Compare this with a smaller receptive field. Let me keep my drawing here. Let's compare this with a smaller receptive field. With a smaller receptive field, if you want to get more sensitivity, oh, it says it right here, smaller receptive fields, more sensitivity, you need less convergence. So we have a green neuron here that goes to its own second order sensory neuron. We have a yellow neuron here that goes to its own second order sensory neuron. We have a purple first order neuron here that goes to its own second order sensory neuron, which means it's going to go to its own third order. So we're getting more neurons going to the same place. More neurons to the same place. More neurons means more information means more sensitivity. Small receptive fields mean more sensitive areas. We have small receptive fields on places like our hand and our face, very, very sensitive parts of our body. This allows us to have 2-point discrimination. So if you have the same, if you poke us with the same distance, with a compass, for instance, at the same distance, you can feel the difference between those 2 points than, say, somewhere on your back. We've talked about We've talked about free nerve endings. We've talked about complex neural receptors. And now here's our third type, a special receptor cell. Specialized receptor cell is where you have a neuron, and it's not necessarily pseudo-unipolar. This should actually be bipolar here, because it's for a special sense, where you still have your first order sensory neuron, but before your first order sensory neuron, you have another cell that is not a neuron. It's usually neuroepithelium. That is our special receptor cell, specialized receptor cell. These are neuroepithelial cells that release neurotransmitter onto the first order sensory neuron. These are special because they're very specialized in terms of what they detect. We have Merkel cells in stratum basale that detect light touch, gustatory cells that detect taste, and let me say this goes on to a pseudo-unipolar. These all go to bipolar because they're special senses. Gustatory cells for taste, those are ones in your taste bud. Hair cells are in your inner ear for hearing and for vestibular sense, your sense of balance, head movement. And photoreceptors in your retina, the back of your eye for vision. These are all neuroepithelial cells. An example of why you need something special here is, for instance, hair cells, what's pictured here, they have stereocilia. Stereocilia help detect fluid movement by bending in a certain way. And that fluid movement comes about from moving your head or from sound waves. So those are our three types of structural neurons, structural sensory neurons, different receptor types. In the coming 2 videos, we'll go over in more detail what's going on in your inner ear for vestibular sense and for hearing, and also in the 3rd video with your eye and how we see things. So I'll see you in the next few videos. Please let me know if you have any questions. Yeah, take care.